are going to make you understand the immunology behind organ or graft rejection. So, before getting into that part, let's see what is organ transplant. Transplantation is a term used to transfer cells, tissues, or organ known as graft from one place to another or from one individual to another. Alexis Carrel reported the first systemic study of transplantation in 1908. He interchanged both the kidneys in series of 9 cats. Some of those receiving kidneys from other cats maintained urinary output for up to 25 years. All the cats eventually died. The experiment established that a transplanted organ could carry out its normal function in the recipient. The first human kidney transplant attempted in 1935 by a Russian scientist failed due to there was a mismatch of blood type between donor and the recipient. This incompatibility caused almost immediate rejection of the kidney, and the patient died without establishing renal function. The rapid immune response experienced here is termed as hyperacute rejection. It is mediated by antibodies and will be described later in the video. The first successful human kidney transplant, which was being between two identical twins, was accomplished in Boston in 1954.
Now, let's look at the terms used for different types of transplant. First is autograft, in which self-tissue transfer from one body side to another in the same individual takes place. Example is kidney transplant. Second is isograft, in which tissues transferred between genetically identical individuals. In humans, is the case of monozygotic twins. Third is allograft, in which the tissues transferred between genetically different members of the same species. The last one is xenograft, in which tissues transferred between different species. Example is the graft of baboon heart into human. In this video, we're going to discuss about allogeneic graft rejection. The graft rejection process occurring in the same species. There are two pathways of the rejection. First is the direct pathway, which is also called as T-cell mediated process. The second is the indirect pathway, which is also called as antibody mediated process. So let's first have a look at the direct process.
In this process, tissues are antigenically similar, are said to be histocompatible. Such tissues do not induce an immunologic response that leads to tissue rejection. Tissues that display significant antigenic differences are known as histoincompatible and induce an immunoresponse that leads to tissue rejection. It is mechanism by which the recipient's T cell recognizes the donor's MHC molecule without any process of recipient's APCC, therefore known as T cell mediated mechanism. This process can be divided into two parts: sensitization phase and effector phase. First, the sensitization phase. The antigen reactive lymphocytes in the recipient's lymph nodes proliferate in response to the donor's allogens. In this steps, the CD4 plus and CD8 plus must proliferate vigorously in response to the foreign antigen. The CD8 plus will be recognized by MHC1 molecule, which are present on the surface of all the cells. Then will induce a cytokine mediated response by TC cells in order to kill the graft, which are identified as foreign antigens, which will in turn creates a cascade of reaction such as coagulation, inflammation, and thrombosis. Now, thrombosis is a process by which the artery gets clogged, which is providing blood to the graft. And this cascade will eventually lead to graft ischemia. This graft ischemia is caused due to the low supply of oxygen as the artery which is supplying blood to the graft is clogged. Now, if CD plus 4 will be recognized by MHC2 cells, then it will cause a delayed type hypersensitivity reaction known as DTH reaction. In some organs and tissues graft, such as grafts of kidney, thymus, and pancreatic islets, a populated donor of APC is called as passenger leukocytes, has been shown to migrate from graft to its regional lymph nodes. These molecules, these, pans, these passenger leukocytes are dendritic cells, which express high levels of class 2 MHC molecule because passenger leukocytes express the allogenic MHC antigen of the donor graft. They are recognized as foreign and therefore can stimulate an immune activation of T lymphocytes in the lymph node. Recognition of the allo antigen expressed expressed on the cells of graft induces vigorous T cell proliferation in host. This will cause the secretion of IFN gamma, which is a soluble cytokine and which activates macrophage to destroy the graft cells. Now, in the effector phase, the recipient sensitized cells will mediate the cell destruction. The hallmark of graft rejection involving cell mediated reaction is an influx of T cells and macrophages into the graft. During the delayed type hypersensitivity reaction, in which cytokines produced by the TDTH cells promote macrophage infiltration, and also IFN increases the class 2 MHC recognition as well. The CD8 plus cell by cytokines and CD4 plus cells do their pathway by macrophages. Now, in this slide, we can uh, see autograft acceptance and autograft rejection. Now, in the first picture of autograft acceptance, from day 3 to 7, revascularization process is happening. That is, the graft is transplanted into the recipient's body, and the blood arteries are or the blood vessels are trying to reach the graft. In the second picture, day 7 to 10, healing process is going on because now the recipient's body have accepted the graft into itself and trying to heal it. Now, the third picture is from day 12 to 14, known as resolution, because we can see that the graft has been accepted by our body and it is resolved into the body as its own. Now, the opposite reaction is autograft rejection. Now, in this first picture, we can see that graft is trans being transferred to the recipient body, and this process is also known as revascularization, which happens from day 3rd to 7th. The second picture is cellular infiltration. In this, the blood vessel or blood artery try to reach out to the graft in order to give its nutrition. In the third process, as we can see, there are numerous blue colored cells present around the blood artery. They are neutrophils. They identified the graft as an foreign antigen and they induced an immune response, which in which caused necrosis. As we can see in on day 14 and onwards, the tissue or the graft went into necrosis and it died. So therefore, autograft rejection took place. This is how a graft is rejected in the direct process.
Now, we're going to discuss about indirect pathway. So in this, allorecognition by the TS cells occurs which are acquired by the host APCs and therefore presented by the MSC class 2 molecules. Some antigens and phagocytized graft cells appear to enter in the MSC class 1 pathway presentation and are indirectly recognized by the TC cells. This phenomenon is called as cross-presentation. It is known as cross-presentation because the MSC class 1 pathway is going to get involved in it. And since the indirect pathway is supposed to be MSC class 2 pathway. Now, generally, this pathway is triggered by trauma from transplant itself and can occur during the procedure with release of foreign proteins from the graft that can be taken up and proceed further by recipient APCs, resulting from the pro-inflammatory environment. The recipient antigen presenting cell, that is your APC, which are MHC class 2 molecules on them, will recognize the CD4 plus cells and the TCDS cells will be activated. Now, since the TS cells are activated in this process, it will in turn activate B lymphocyte. And we all know that B lymphocytes are majorly involved in antibody production. So therefore, in this process is also called as antibody mediated hypersensitivity reaction because in this process, the antibodies are produced independent. Now, in this reaction, uh, as antibodies are produced, plasma cells are activated, and antibodies are released in the graft. These antibodies, which are dispersed in the graft tissue or organ, will act as a flags of destruction for the graft cell. These antibodies will be recognized by macrophages and cause endothelial injury, and in turn will cause graft ischemia because the artery which is supplying blood to the graft tissue is blocked. Therefore, there is no supply of nutrition as well as oxygen is not there. This will cause ischemic condition and is known as graft ischemia. This eventually will kill the graft cells and cause graft rejection. So as of now, in this process, there is a major role of antibodies which are produced by the B lymphocytes and they're further destroyed by the macrophages.
the direct and the indirect process which we just saw. So as we can see, the APC and the TS cells are interacting with each other, and the TS cells can be further so, seen as four different types. Now, the first three types are of direct process, and the last type, that is B cell, is of indirect process. In the first type, we can see that TDTH cell is present. It is secreting IFN gamma and IFN beta. Now, the IFN gamma secretion will activate a macrophage, and the macrophage will secrete lytic enzymes. Now, the lytic enzymes are going to rupture the cell membrane of the graft cell and eventually causing the graft rejection. Now, the IFN beta cells are involved in cytotoxicity. These cytotoxic cells are going to rupture the membrane of the graft and going to kill the graft. Now, second is the CD8 plus cells, which are the TC cells. Now, the CD8 plus cells are recognized by MHC class 1. These, when they are recognized, they are going to damage the membrane and then cause graft rejection. The third one is CD4 plus cells. These CD4 plus cells are recognized by MHC class 2, and they are also going to do the same. They are going to cause membrane damage and thus causing the graft rejection. So this was the process of direct method.
So now, as we can see that in indirect pathway, the TS cell is going to activate the B lymphocyte cell. And this B lymphocyte cell will interproduce antibodies which will go and sit on the surface of the graft cell, and they are recognized by the either NK cells or macrophages. This recognition is done by Fc receptors. Now these Fc receptors will recognize antibodies which are present on the graft cell or graft tissues, and then macrophages will kill the graft tissue. So this is how we came to know about the two pathways, that is your direct pathway and indirect pathway. And by both the processes, graft is rejected.
So now let's just see some facts of organ donation or organ transplantation. There are instances in which an allograft may be accepted without the use of immunosuppressive measures. Obviously, in the case of tissue that lack alloantigens such as cartilage or heart valves, there is no immunological barrier to the transplant. However, there are also instances in which the strong predicted response to allograft does not occur. There are two general cases in which an allograft may be accepted. One of when the cell or the tissue or grafted to a so-called privileged site that is sequestered from immune system surveillance. And the second is when a state of tolerance has been induced biologically, usually by previous exposure to antigens of the donor in a manner that causes immune tolerance rather than
Now let's have a look.
sensitization in receptor.
Now let's have a look at challenges faced during organ transplant surgery. So during organ transplant surgery, doctors are always facing and racing against the clock, and time is not on their side. Also, during the surgery, there are several precautionary and preventative measures are taken into consideration. But still, the time and the major problem is that keeping organs viable outside the body before being transplanted. Depending upon the organ, the time range can vary from few hours to a half a day. For example, liver has a viability of 12 hours, lungs 6 to 8 hours, and kidney 24 to 36 hours. These are the commonly transplanted organs. But when it comes to heart, it is quite difficult because it loses the viability the fastest when stored in cooler. Also, we are only 6 hours to work with because the longer it will stay outside the body, it is more difficult to transplant. But the new medical device industry known as Transmedic is making a difference. The company has developed an organ care system known as OCS. It is an integrated system that allows transplant teams to keep organs alive, beating and breathing between the donor and recipient instead of storing them on ice. After the heart is removed from the donor, it is attached to what is called as organ care system or OCS. It kept it warm, it fed nutrition, it circulates the blood. Basically, it kept it alive. The OCS heart and OCS lung system are CE marked and clinically used in transplant centers in Europe, Australia, and Canada. Hence, heart in a box is going to replace the old therapy and nothing to do with the ice anymore.
So Now we will see the statistics of organ donation in India. Every year, August 13 is celebrated as Organ Donation Day. The day is celebrated to motivate and encourage people to donate their organ and to understand the value of organ donation in life of an individual who needs it because lakhs of people die every year waiting for an organ and every 12 minutes one more patient is added to the waiting list. According to a survey, every year in India, about 500,000 people die because of non availability of organs, 200,000 people die due to liver disease, 50,000 people die because of heart disease. Moreover, 150,000 people await a kidney transplant, but only 5,000 amongst them get it. Despite India being the world's second most populous country, it has a pathetic diseased organ donation rate of just 0.26 per million population compared to 26 in US, 35 in Spain and 36.5 in Croatia. India is struggling with an acute shortage of organs for transplantation. It is estimated that more than a million people suffer from end stage organ failure, but only handful of 3,500 transplants are performed annually. Awareness of organ donation is therefore the only way out of this depressing scenario. The more potential donors there are, the more the likelihood of organ becoming available to save our lives. And now we would like to end our video by quoting,
The idea is not to live forever.
but maybe to help another live a little longer. Thank you, everyone.
Thank you so much.